Good afternoon. Very happy to see so many familiar names in the people's list. I'm trying to get the Twitter feed to go as well, but apparently there is an issue today. So if you're following, oh, there we are. Twitter is live as well. Okay. Welcome to Worldwide Neuro. My name is Tim Vogels. I am today still an associate professor at the University of Oxford. It happens to be my last broadcast in this function. This is the last time I'll be hosting an Oxford Neurotheory seminar because I'm moving to the Institute of Science and Technology Austria in Klosterneuburg by Vienna. And today, as my last guest, I welcome Phillip Berens. Before I introduce Phillip, very briefly, you already know by now the functions of Crowdcast. There is a chat bar on your right that you should use at your leisure to ask questions, to interrupt the speaker, or to allow me to interrupt the speaker on your behalf. If you have more urgent or more complex questions that you want to come onto the screen for, ask a question via the Ask a Question function in the bottom. And then, of course, follow us via Twitter or join our email list. You can do that by clicking the button in the bottom of the screen there as well. And then I wanted to quickly tell you that we are starting a franchise of kinds. So if you have friends from other fields that think Worldwide Neuro is cool, we're allowing other people or other branches to come on board quite quickly with Worldwide whatever. So we have that in the works. If you need any help with that, let us know. And then today's speaker, Phillip Berens, told me his CV looks as follows: Tübingen, 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 Tübingen. So Phillip did his PhD in Tübingen with in the Bethke lab, I should say, and then did a postdoc with Tom Euler. And in between, I think he was also with Andreas Tolias in the States, but really split his time to also be in Tübingen in Tom Euler's lab, I guess, or in Bethke's lab. He received the Bernstein Prize in 2015 and subsequently started his own lab also in Tübingen in 2016 and has been a professor there, a Heisenberg professor, since 2018 and remains in Tübingen and is looking actually for postdocs. So if you're interested in joining his lab, listen carefully to the next 60 to 90 minutes. Without further ado, Phillip, thank you for coming. The floor is yours.
Yeah, thank you, Tim, for inviting me and for the honor of being the last speaker of your Oxford legacy. I think the Worldwide Neuro Talk series is really a major game changer, I think, for neuroscience exchange across the world. And we've also been setting up a small series here, and I think it's really cool to have that kind of exchange. So
thank you.
Let me share my screen now. Okay, I hope now you can see my slides.
Yes, we can.
Okay, yeah, I presume since nobody's complaining that this works. So for the slides, I shortened the title a bit because I think it was a bit too long. But so the neutral title is just Towards Hybrid Models of Retinal Circuits, and I'll explain in a minute what I mean by hybrid. So at the beginning, I would like to take a little bit of a step back and ask ourselves, so why do we use model in science at all? if you reflect on that question and also maybe look a little bit into the philosophy of science, I think the function of models in science is basically twofold. On the one hand, we want to use models to summarize our understanding of the system. If you want, it's a sketch of a system that allows us to write in a concise form, in a mathematical form, what we know about it. And on the other hand, it allows us to create predictions from that summarized description to test our understanding. So ideally, we can create predictions that can be tested experimentally. Otherwise, it wouldn't be science. And then we can actually go about and do that in the lab. In neuroscience, we mostly have kind of two kinds of models of neurons. On the one hand, we have models that are inspired by machine learning or statistics, and on the other hand, we have more mechanistic models that are inspired by the biophysics of the underlying systems. So on the side here of machine learning and statistical models, you can maybe even start with the most simple ones, something like a linear nonlinear Poisson model or a spike-triggered average model that describes the relationship in visual neuroscience between some set of visual stimuli and some set of spike responses by a linear function that is passed through a simple nonlinearity and some added noise. So this is essentially it's within the framework of generalized linear models from statistics, and provides a statistical map of the input-output behavior of the system. On the other hand, we sometimes use mechanistic models to describe neural systems. Like in this case, this is one of our papers, but of course this goes back to the famous work by Hodgkin and Huxley. So here you can see a dendritic branch of a horizontal cell in the retina, and then we have these segments of membrane compartments that are modeled by Hodgkin-Huxley-like equations, and these can be used to describe the activity of the neural system on a mechanistic and biophysical level. So what are the various advantages and disadvantages of these two approaches, and how do they work? So on the side of these machine learning and statistical models, the goal of such models is to maximize predictive performance. So given some set of visual stimuli, for example, the goal of such a model is to predict as well as possible the output of a neuron, the spike train. What we put in is typically some form of training data. So we put in a set of recordings of which we know the stimuli and we know the spike trains, and then we try to learn or infer the parameters that map the input to the output. And in addition, we put in an inductive bias by choosing the model class, by choosing the set of functions in which we learn. Typically, or in most cases, the equations that underlie such models are relatively simple. So for example, in the case of the linear nonlinear Poisson model, it's really just linear regression with another output function. As a result, and because of all the great engineering that has been going on in machine learning in the last couple of years, oftentimes such models are relatively easy to fit to data. So by easy, I mean not that it's not challenging to do that, but it's comparatively clear what one needs to do. And as a consequence, such models have been also very successful in predicting neural activity. So this is from a paper by the group of Matthias Bethke, Santiago Cadena, and it's just one of many examples where they took a machine learning model here, a VGG network, they passed visual stimuli through that model, and then took the feature maps that resulted from this model and combined them with a linear readout, nonlinearity, and some noise model to predict spike counts in McCarthy. And such models, right now at least, are really the state of the art in predicting the responses of neurons in many systems. So what about mechanistic models? But the problem with these machine learning and statistical models is that their interpretation is not easy, or sometimes can be rather difficult. So if you have, for example, such a neural network model of a neural system or of a neuron, you really have to do almost as much work to understand what's happening inside the neural network as you would have to do for the brain itself. So what about mechanistic models? So here, I would say the goal is not so much to maximize the predictive performance, but it's rather to further our understanding of the underlying biophysics. What we put in is all our knowledge about the system. So all the biophysical mechanisms that somebody has measured before or that we assume to be known. The big advantage of that is that the parameters of the model are typically easily interpretable. So, for example, if in a Hodgkin-Huxley style equation, you have a parameter for the channel density of sodium channels, that is something that really relates very in a straightforward way to a quantity that one can reason about. On the other hand, everybody who has worked with such models, the underlying equations are often complicated. They involve differential equations. They cannot be solved in closed form. They have to be simulated, and thus are very hard to fit the data. Of course, what one would want is the best of both worlds. So we would like to obtain models that on the one hand have high predictive power, and on the other hand are biophysically plausible and have interpretable parameters in a biophysical sense. So we want biophysically realistic neuron models, for example, through some set of Hodgkin-Huxley style models. But we want to be able at the same time to perform parameter inference from relatively standard stimulus response measurements and achieve good predictive performance. So the goal of achieving high predictive performance is really something that we also want to keep. And finally, in an ideal case, we not only get that, but ideally we also get an uncertainty representation of the parameters that allows us to detect parameters that were not constrained by our inference procedure or do other forms of uncertainty-based reasoning with this information. So what I would like to show you today is two case studies that we did in my lab over the last 2 years or so. And this is what I call hybrid models of neurons. So these are models that have biophysically interpretable components, but at the same time, they are optimized to really maximize predictive performance. The first one is a more simple case. It's a case of a feed-forward linear nonlinear release model of a single bipolar cell in the retina that also allows for uncertainty quantification. And the second one will then be a circuit model of all the bipolar cell types and including inhibitory neuron feedback in the retina. Okay. So where are we in the brain? What's the model system that we study? This is in this talk. This will be mostly the retina. The retina, as you know, is in your eyes. It's a sheet of tissue that takes up light and transforms light into first electrochemical potentials and then later sends them to the brain. So the light is taken up, as you know, by the photoreceptors. It's forwarded to the bipolar cells, which are the main excitatory feed-forward neurons of the retina, and then here relayed to the ganglion cells that send it to the brain, the information to the brain. And the processing is modified here by two populations or two large subclasses of amacrine cells, inhibitory interneurons, that either mediate kind of lateral interactions or also horizontal interactions in this very dense synaptic plexus called inner plexiform layer. So what will the first model be about? The first model will model the relationship between visual stimuli coming in, being transformed by some sort of linear filter, applying some sort of nonlinearity, and then, and this is the part where it becomes a bit biophysical, passing that through a synaptic release mechanism that tries to accurately capture our current understanding of how the release mechanism works. And then we also model the imaging process in a simplified form. This first project was, I have to say, a collaboration with Ben James and Leon Lanyado from the University of Sussex in the UK. And the data we got from them were recorded in fish bipolar cells. They were recorded with two-photon imaging of an indicator called igloo sniffer. Igloo sniffer is it's a protein that changes its fluorescence in response to concentration changes of glutamate. And glutamate is the primary neurotransmitter that is released by the bipolar cells. And if I'm not mistaken, in this image of a bipolar cells, this would be kind of here. What they did in the experiments, they played this series of binary noise stimuli flickering between bright and dark light very rapidly, and they obtained imaging trace that looked like this. So you can see very nice transients sometimes happening, and the amplitude of these transients also varied. And what they showed in a paper that was, I think, published last year in Nature Neuroscience is that the nature of this release is actually quantal. So that these there's some form of smallest event, and then all the other events are multiples of that, presumably because discrete number of vesicles is released at any given time. And one can then go and process this data to obtain really the number of vesicles that's released in any given time point. And we work with these traces. What one has to note is that these traces that are kind of converted into quantal events, they are very noisy due to the statistic of this kind of quantal release procedure. The model outline, just to orient you again, is this. So light stimuli are taken, passed through a linear filter. This is kind of, if you want, modeling intracellular calcium concentration. This is passed through a nonlinearity that converts the calcium concentration into a driving force on the release from the ribbon. So the ribbon is a synaptic structure. I'll explain that here on the next slide. So here we have an EM image of the ribbon, and the ribbon is a synaptic structure where some vesicles are docked here very close to the membrane. They are releasable immediately, so this is called the readily releasable pool, and this release process is triggered by an increase in intracellular calcium. In addition, there's this ribbon structure, and a lot of vesicles with neurotransmitter are docked to that ribbon structure. This is called the intermediate pool. They can replenish the readily releasable pool fairly quickly. And then there's, in addition, a lot of vesicles around in the cytoplasm that then dock to the ribbon and are moved towards the readily releasable pool more slowly. But think of the ribbon as a conveyor belt that kind of shuttles vesicles closer to the membrane for them to be released rapidly. And you find such synapses. These are really specialized synapses that allow for very quick, strong release of glutamate. And you find them in many sensory organs, not only in the retina. So you also see them in the hair cells and so on. And they are often also very specialized for their particular application. So how do we describe what's happening in this in the ribbon? I just described it to you in words. What is happening? How do we describe that in terms of math? This is summarized here, and I don't want to go through all the details of these equations, but basically we model the vesicle release in a way that allows for correlated vesicle release, so multiple vesicles to be released at the same time. And we do that by this kind of better binomial distribution. Then we model the movement of the vesicles to the dock. This is works very similar, but we cap the release by the maximum size of this intermediate pool, and then we replenish we assume that this pool of vesicles in the cytoplasm is virtually infinite, and we replenish our intermediate pool at a somewhat slower timescale. Now, we would like to infer all the parameters of this model. This is basically, these are the parameters of the nonlinearity and of the linear filter, and then basically of the transition rates and the pool sizes in the rhythm. How do we do that? So this is not so straightforward, and we want to do that in a way that we get, in the end, uncertainty estimates. Now, if you hear uncertainty estimates, you think Bayesian inference, and what you, of course, all know is Bayes rules, right? So you have the posterior, the probability of the parameters given the data. This is proportional to the likelihood times the prior, so the probability of the data given the parameter times the probability of the parameter, a priori probability. Now, for such a model, as I just described to you, which is based on simulations, so you can, given a set of parameters, you can very easily generate a forward pass of the model and simulate a corresponding data trace. But what you cannot easily do is compute this likelihood factor here. So what you need to do instead is resort to special techniques that have been gaining a lot of traction in the last couple of years, called likelihood-free Bayesian inference. And this is also what we do here. So in this particular project, we actually use a very simple version of that, where we sample from a prior. So this is the blue outline in the back, some prior distribution. We sample a set of parameters, then we run the model with that set of parameters and evaluate it. We extract some summary statistics that we think are important. So let's say the overall number of events, the distribution of the size of the events, and so on. And we evaluate a loss function that measures how good the summary statistic, the extracted summary statistic for the current set of parameters, how well that matches our data. And we do that many, many times, let's say 10,000 times, and we accept the best sets of parameters. And then we can compute an explicit Bayesian update formula for the distribution of the parameters. And then we run this whole iteration again, and we do that a couple of times until it has converged. Now, how does that look for the model that I just described to you? So our linear, nonlinear release model. So the data, these are just two samples of the data. You can see they are, although they were the neurons shown the same light stimulus, they're quite different. When we fit our model to it, we see that it relatively rapidly converges. So the inference is possible. These are all or some of the model parameters. The blue trace is really kind of the round by round inference outcome. And then in light blue, you can see the uncertainty in the estimate of the parameters. You can see that actually many of the parameters are really well constrained, but there are some, like this release probability here, that is less well constrained by the data. So with these inferred parameters, how do simulations look? And if you look at them, you say, okay, well, it kind of looks different, but overall the peaks seem to be roughly at the right place. So how close is this really to the data? So we evaluated our loss function that for various repeats of the experiment among each other, and then you get a value of about 4 or so. And then for the data compared to the model, and then you're a bit worse, but not so much. We also compared that to a simple baseline model, in this case just a simple GLM, and you can see that mostly models much smaller events, and the loss is also much bigger. You can visualize that further. If you look at how many vesicles are released at any given instance, then what's quite obvious already from the plot is that the distribution is such that in the GLM model, a simultaneous release of multiple vesicles is underestimated quite a bit. Now, what is such a model useful? Well, we can also check whether this model captures properties of the neurons that are not used for fitting. So, for example, in this case, we here show the number of vesicles released compared to the temporal precision with which the events occur. And what Ben and Leon found in the other paper that they did is that in data, there was a trend where events that had more vesicles released also had higher temporal precision. And that is something that is also captured by the model. And furthermore, if we apply kind of a step pulse to the model, then the cumulative release goes up like this, and this really closely matches data that has been measured experimentally as well. So this first model now provides a good account of bipolar cell activity in the fish, and combines so much of those cells' details are summarized in that linear filter. So almost the entire cell, what's happening on its dendrites, what's happening in the soma, on the axon, and so on, is all summarized in that linear filter. The only part where we put in some biophysical detail is actually the ribbon. There we provide a stochastic mathematical model for it, and we do that because we would like to understand how important is that this kind of ribbon structure in shaping the output of the bipolar cell. Now, in a parallel project, we also showed that one can do that with a much more complicated model. So where we actually build explicit biophysically realistic multi-compartment models of the photoreceptors, of the whole dendritic tree, and the axon. And so in total, this model would have about 23 parameters corresponding to the density of various ion channels, such as T and L-type calcium channels, and so on. And also, again, the release cascade and the membrane resistance, and so on. And what the problem here is that many of these parameters are not measured in any specific animal that you're looking at. So in the past, of course, they have been measured. And then we use more advanced inference techniques from Jakob Macke's lab to infer these parameters for such a complicated model and fit that to data and obtain uncertainty estimates. And this is also possible. So if you're interested in the details of this work, this was done by also a PhD student, Jonathan Oesterle, then a preprint of that is available on bioRxiv. Okay. So what I've showed you now is that we are able to build and fit a feed-forward model of with a linear, nonlinear stage and explicit biophysically motivated release stage that is able to predict the activity of a bipolar cell reasonably well. So now Tim told me that you would maybe ask some intermediate questions at some point, and I think now would be a good point to do so.
There haven't been any so far, so otherwise I would have interrupted you.
Okay, great.
I think, yeah, feel free to pop me questions into the chat box if you want, but seems like everyone's...
Then we'll just go on. So now I come to the second part of the talk, where I would like to present to you our latest work on a circuit model of all bipolar cell types, including American cell feedback. This is motivated by joint work with Katrin Franke and Thomas Euler that we published some years ago, where we showed that the influence of American cells, right, these two classes of American cells, that what they what this does is that they act to decorrelate the different bipolar cell types. I can briefly explain to you in the data shown here. So this is the stimulus that we use. We always use that stimulus to study retinal cell types, so it's kind of our signature stimulus. Feel free to use it in your experiments, of course, as well. It's not protected by anything, no patent or whatever. It has this kind of on step here, light step in the beginning, then it has this rapid increase in frequency and then a rapid increase in contrast. So if you look now first at the light gray responses, then these are two off cells, these are two on cells. And what you will notice is that in this case of the light gray responses, the stimulus was only shown as a very small disk, just covering the receptive fields of all these neurons. And what you can see is that under these local conditions, the different off bipolar cell types respond very similarly, and the different on bipolar cell types respond very similarly. And you can also look at that in this correlation matrix between all the different bipolar cell types. So here, this is almost like a perfect correlation. And the same, this is the off types, and these are the on types. They also have a very high correlation to the local stimulus. If you make the stimulus larger, then you can see here immediately that the average correlation level between the different types changes and goes down overall. And you can also see that here in these responses, immediately that they become more diverse. So here, for example, this really changes quite dramatically. This becomes much more transient, and so on and so forth. So, to explain that, we also did all the necessary pharmacology experiments blocking the different AC populations. By we, in this case, by the way, I mean Katrin. And we can show that it's really what happens when you use this larger stimulus, that you recruit in the surround more amacrine cell populations that really mediate these effects. And if you're interested in the details of that, this is all published. Now, we would like to obtain a model that captures all these effects. So what is missing from our first simple model here of the vertical pathway of a single bipolar cell responses is the feedback that is provided by the local amacrine cells. And this is, so I don't have sorry about that, I don't have the pictures here, but this is joint work between Cornelius Schröder, who is a finishing PhD student in my lab, and David Klint, who is a PhD student in Matthias Bethke's lab. Now, we take the same model as before. We simplify it a bit because we take out the stochasticity, and then we feed the output of that model. We feed that into these feedback circuits here of amacrine cells. These orange amacrine cells, they are horizontally connected across, vertically connected across the inner plexiform layer, and they also provide feedback to other amacrine cells. And then these global amacrine cell populations that just provide inhibitory feedback to bipolar cells. And of course, so this model has really like a lot of parameters. So on the one hand, it has all the parameters of the feed-forward pathway for all the different types. And it has all the parameters for this feedback pathway, and we include actually quite a diverse set of potential amacrine cells motivated by connectomics data set from Moritz Helmstedt. So how do we model that on the mathematical side? Well, the activity or the activation function maybe of the bipolar cell is a function of the input, and then it's inhibited by the local and the global feedback. And this activation here then drives the simplified model of the release. So again, this release from the readily releasable pool. There's some refill going on from the intermediate pool, and the intermediate pool is refilled at every time step. The amacrine cell. So here's this in the output of the bipolar cell, we have this nonlinearity of the release, but the rest of the model is linear with kind of static nonlinearities. So the output of an amacrine cell is an integration over some time window, some amacrine cell kernel here, of the aggregated input from all the bipolar cells times some weight matrix. And there's local feedback to the amacrine cells. And then this output of the amacrine cells is combined to provide local feedback to the bipolar cells and global feedback to the bipolar cells. This model, Cornelius and David then implemented in PyTorch, and also used the inbuilt inference engine of PyTorch to fit the parameters. Now, we tested the model, or we trained the model on a dataset containing these TERP stimulus under local and global conditions, because this is important to constrain the model to realistic activations where the amacrine cells are actually in action. And in green, you always see a simple control model, a linear nonlinear model, and in blue, you see kind of the you see an LSTM model that is very agnostic to the actual circuit, but has as many parameters as the bipolar cell network model, and is very flexible. It's a pure machine learning model. And what I'm showing you now is the training performance. So what we see is that basically the LN model, numerically kind of okay, models the data, but not spectacularly so. And both the BCN and the LSTM model, they almost perfectly learn the structure in the data. And this is also what we see here in when we look at some of the actual traces. So here the LSTM model really just perfectly fits the data, and the BCN model does it almost as well. I remind you, this is training data, so this is not particularly surprising. It could be that all of these models are horribly overfitting. What is interesting, though, to note is that the LN model has very clear deficits. So, for example, here, it's able to model this kind of step response, and it overall captures when the neuron fires at all. But what it doesn't model is this very rapid release right after stimulus onset and this adaptive process. Or also here, the adaptive process throughout this increasing speed is not modeled by the linear nonlinear model. And you can also see that here. But the other two models, they can capture that. Now for the more interesting case. We tested the model on several data sets that it was not trained on, but I will show you only the test performance here on a set of natural movies. I have to say, the model so far only captures all the temporal activity and the temporal relationships. So what we did is we took the responses of bipolar cells to a natural movie, and we put a rough Gaussian receptive field on top of that natural movie, and filtered with that Gaussian receptive field, filtered the spatial structure with some assumptions about the surround to obtain actually such a simple time series here. And in response to that time series, one particular bipolar cell type responds like this. So this is now really test data. This is actually quite challenging, I would say, test case, because it's not only data that the model hasn't seen, but it's actually data from a different distribution. And so it's a little hard to see, and the data is also quite noisy. But when we quantitatively compare the model performance, we can now see that the bipolar cell network model actually has a much higher correlation than the linear nonlinear model. So here the colors correspond to on and off cells. So the blue ones are on cells and the red ones are off cells. And we can see that for no matter whether it's an on and off cell, they are all basically above the main diagonal here, so which indicates that the bipolar cell network model is really better than the LN model. And interestingly, it's for this test data, it's now even better on average than the very flexible LSTM, indicating that for the LSTM, there's actually some overfitting going on. Okay, so now we established that our bipolar cell network model performs as well as really like a very flexible model without any biophysical knowledge put in, that should be able to learn all the structure in the data. But whereas this model is very hard to interpret because it's a black box model, we feed in stimuli and we get out responses, but we don't know about anything about its inner workings. While with a bipolar cell network model, we can now do in silicio experiments. So for example, we can mimic the effect of blocking certain amacrine cell populations using pharmacology. So for example, in one experiment, Katrin applied a drug called strychnine, and that blocks the effect of the more local amacrine cells, providing vertical feedback. When you do that, and you can first look at the traces behind, these are kind of the measured experimental data. When you block the local amacrine cells with strychnine, you can see that basically there's more release that goes kind of under the baseline. So there's stronger sub-baseline release in both cases. This is also something that, and this is captured also by our model without having ever been fed of data from these pharmacological conditions. And this is also something that we can quantify for relatively scarce data because the experiments are very difficult. So in the model and in the data, the overall trend, so this kind of tonic release index, this quantifies this effect that you can see here. And in the model and in the data, this effect really has kind of the same direction. I think so. The magnitude is somewhat different, but I think this is not surprising because there's, of course, in the data there's also some noise and so on, which is not present in the model. Further, we can study the structure of the learned connectivity matrix, and I here just show you an aggregated matrix. So just to explain that first, so here it's bipolar cells, local amacrine cells, and global amacrine cells, and then these yellow lines, they always indicate all the cell types in that respective class that are classified as on cells. And then we average over all the weights between bipolar cells and local amacrine cells, and between bipolar cells and global amacrine cells, and so on and so forth. And when you just visually compare the structure of this connectivity matrix in the model and the connectivity matrix that was extracted from the EM dataset generated by Moritz Helmstetter and Kevin Brickman, then you find that these provide a relatively good match, at least visually. There's caveat, so in the EM data, this was one of the first big EM data sets to be generated, and for ease of tracing the membranes, they at that point didn't stain for synaptic markers such as rhythms. And so therefore we cannot really distinguish simple contacts from functionally active contacts or synapses. Okay. Now, I said earlier that we would like to use the model. Yes.
There was a question. I'll just read it to you. Can the underlying voltage model, I guess the BC terminal generator, add the possibility of spikes or spikelets? What does that do to the response predictions?
No, it doesn't. There's at this point there's no kind of all or none model, but that would be actually an interesting test case that one could try to include, I presume. Probably Cornelius is fervently shaking his head in the background somewhere. But so in bipolar cell terminals in Tom's lab, Tom Barden's lab, they have been shown to be able to generate spikes. Or also when he was a postdoc with Leon earlier. And that might be actually an interesting direction to extend that model, to test what they, whether this actually is necessary to improve this modeling here. What I have to say is the way we fit this model is actually not on individual traces, but it's on averages of many cells. So the cluster kind of averages or cell type averages across many cells and many repetitions of the stimulus. So they're fairly noise free, and any effect of including like this kind of stochastic spiking mechanism at the terminal would average out. But I think it would be an interesting direction.
Thank you.
Sure. All right. So I said in the beginning that we would want to use model to create predictions. And here's a prediction. So if we look at the parameters of the model and we look at all the different bipolar cell types, what our model predicts is that there's actually strong differences in the ribbon properties between bipolar cell types. This is the readily releasable pool capacity, and this is the transfer rate from the immediate pool to the readily releasable pool. And you can, and all the colors here correspond to they correspond to model instantiations being trained in parallel. You can see that across all these model instantiations, quite robustly, you can distinguish many of the different bipolar cell types. So basically the different colors here by these two properties. This is maybe not entirely surprising because the readily releasable pool here is also correlated with the transients of the responses. So the higher the capacity of the readily releasable pool is, the more transient the responses are. So it allows it to release a lot of neurotransmitter at in the beginning very rapidly. And so now the question is that a plausible prediction or not, and how can that be tested experimentally? So what is known across the animal kingdom and also across different species or different even regions in the eye also is that that ribbon properties can change and are adapted to the specific environment. So for example, if you go, I think, to some shark, they have insanely huge ribbons. So it's not entirely unplausible that something like this could be happening. It's not so straightforward to test the idea, but one would have to do EM and then assign the terminals to respective bipolar cell types and do that in a way that actually really captures kind of the vesicles still being discernible and so on. But so at least I would say it's a genuine prediction of the model, and maybe at some point somebody will be brave enough to test that. So, and with that, I'm already close to the end. So I showed you two hybrid models of neurons. So here first, a feed-forward linear nonlinear release model of a single bipolar cell or single bipolar cell type. And then a circuit model of all bipolar cells, including amacrine cell feedback, that allows us to study the role of different amacrine cells in the model, study the structure of the connectivity matrix and the importance of specific things in the connectivity matrix. And then finally also allows us to generate predictions about biophysical properties of these neurons that can later then be tested experimentally. And with that, I would like to thank all the members of my lab. Much of the work that I showed today were spearheaded and led by Cornelius Schröder, and of course also by all the people who pay for our work, so most notably the Federal Ministry of Education and Research and the German Deutsche Forschungsgemeinschaft German Science Foundation. As Tim already said in the beginning, if you look for a postdoc position at some point in time, please send me an email or contact me on Twitter. And with that, I'm happy to take more questions. And thanks everybody for listening.
I'm the only one clapping, obviously, once again. Thank you very much for your talk. We do have a few questions. Before I pass the mic, so to speak, I was wondering, you kind of move back and forth between fish and mouse pretty seamlessly. Yeah. Do you have to adjust your models, and what were, like, it seemed like even in the second part you had both fish and mouse, or did you just have mouse in the second part?
No, it's not a Faust in the end. The second part was just mouse data, and the first part was just fish data. So, I didn't jump too regularly.
What do you have to do? Like, do they transfer easily, or do you have to adjust the models a lot? Could you use your model from the first half for the second half, and why did you?
This is actually pretty much what we did, at least the structure. So and we were actually in the beginning we were actually surprised how well that worked. So we thought we would have to modify that much more. I think the advantage in the fish, so the experiments that Ben did in Leon's lab, the advantage was that they were scanning really fast, zooming in on individual bipolar cells and individual axon terminals. So they could actually resolve this multivesicular release, this quantal release there. So, and this is what we made this stochastic model for. And then we went to when we went to the mouse data that we had analyzed together with Thomas Euler and Tom Baden and Katrin Franke. We typically anyway work with trial averaged or repetition average data. So it's a little bit different, and it's also more of a population imaging approach. So there we looked at recordings that I don't know, at least covered like small bits of the IPL, not really individual synapses. So there were some differences in the experimental techniques that were used to acquire the data, but at the same time, when we transferred the model and basically took out the stochasticity, we were actually surprised by how easily it can be adapted to also fit the mouse data. And I think the most likely, but all the biologists in the audience may correct me if this is stupid. It's because the structure of the ribbon is fundamentally similar, even if you have to adapt the precise parameters.
Is there any EM data that suggests that ribbons are structurally tuned?
Stay tuned. So we're working with Tom Baden. We're kind of working into that direction right now in fish photoreceptors.
Okay. All right. I have two more questions. One is, can your hybrid models predict or account for contrast or luminescence normalization?
So you mean if we change like I don't know, if we go to much higher light levels or something?
Yeah.
Yeah, this is something that we haven't looked at much in the experimental data, because when you do the two-photon imaging experiments in the retina, you always have the background illumination from the laser, which is fairly bright already. So I guess you could kind of go up from there, but there's nothing that we have looked so far.
Mm-hmm. But in the
model, it would be very interesting.
Yeah. Okay. Then there is a question from Wei Li. Which BC types have the smallest and the largest RRP? And what would an RRP smaller than 1 mean?
Yeah, I think you already commented on that on Twitter, on what these numbers actually are. So I would more look at them in terms of of their relative ordering, I would say. I'm not sure whether I would say like, yeah, this should be a capacity of really of 50 vesicles or something like this. So I think it's really more like the relative ordering that is important here, and I think a pool of vesicles that's smaller than 1, that indeed wouldn't make much sense.
Okay. I've invited Wei to the screen, so he might join us. He has another question, but I'll wait for him.
Can I somehow see that screen as well? Because right now I just still see my slides.
You just have, if you go out of presentation mode, you should be able to see us all, or at least me and the question.
Yeah, but then we have this infinite recursion problem.
Oh no, that's fine. Try to do this.
Okay.
There you go. So a question from, there's Wei.
Hi.
Hi.
Another question is, for different type of bipolar cells, those two parameters K1 and K2 for the vesicle replenishment, do they differ largely, or they're quite similar?
I think that would be also good to look at, but we haven't so far. But I mean, I can ask Cornelius to look into it, and we can send it to you.
Okay. I thought it would be interesting to see whether the replenishment processes will be similar for the different types, rather the RPs differ because, say, ribbon structure or.
So I think, so my assumption would be that we here picked the parameters that most differentiated the different types. So that would suggest that the thing that you are asking about is probably more similar between types, but I would have to look into it to give a precise answer.
That will make sense. Thanks.
Good. Thank you, Wei. And the last question by Tom Baden. How do BC models compare to models in other cell classes, for example, photoreceptors?
Thanks, Tom, for the excellent question. I'll give an answer, and you can say whether you're happy with it or not. I think ultimately the same kind of model structure can also be used to model a photoreceptor. So we could just take this feed-forward model that we have and model release from a photoreceptor. It also has a ribbon synapse. There's only in the outer retina, there's only one type of interneuron, horizontal cells, that also provides feedback to the photoreceptor. So I think the structure of the model would be much more simple, but I think a very similar model could be built or used. So I'm not sure what you had in mind beyond that.
I think that's fine. I don't know what he had in mind either. Tom, if you want to answer, but there's nothing. There's no follow-up. So, he says thank you. So, Philip, I think this is it. Thank you very much for the lovely talk. I will send you a Zoom link for afterwards.
Okay.
But for now, this is goodbye. We'll finish our broadcast here. I thank you all for coming, and remain with the best wishes from Oxford. See you again soon under a different name at the end of the summer for a new Euro-Neuro theory series. With that, thank you, and thank you again, Philip.
Yeah, thanks everybody for coming and listening.
Bye.